Hello everyone. In this video series, we are talking about precipitation reaction. It is the type of immune reaction in which soluble antigen combine with its antibody to produce insoluble precipitin complex, and this complex can be easily detected in liquid or in gel medium. When precipitation reaction occur in the gel medium, it is called as diffusion. Okay, and we have already talked about three important type of technique which is based upon precipitation type of reaction. Number one is single radial diffusion. Number two is double immunodiffusion. And number three is immunoelectrophoresis. So in the first type of a precipitation reaction, only antigen or antibody move towards other partner. Okay, that means in case of single radial immune diffusion, out of the two, antigen or antibody remain fixed or stationary, and other partner move towards stationary partner in the gel or in the liquid medium. So it is called as single radial diffusion because the diffusion occur radially. Now in case of double immunodiffusion, both antigen and antibody are free to move towards one another. So we call it as a double immunodiffusion. In the last, that is immunoelectrophoresis, this type of a precipitation reaction occur with the help of electrophoresis. Okay, so electric current is applied to the antigen mixture and antigen migrate in the gel with the help of electric current. So it is called as immunoelectrophoresis. I have discussed about all these three techniques in detail in my previous video. Now in this video, I am going to focus on next type of a technique which come under the precipitation reaction, that is rocket immunoelectrophoresis. Okay, so this rocket immunoelectrophoresis is also called as Laurell's reaction or Laurell's technique, because Laurell developed this technique to determine the concentration of unknown antigen from the mixture of the antigen. Okay, so if we consider rocket immunoelectrophoresis, or if we compare rocket immunoelectrophoresis with all other technique of precipitation, you can find this is the very simple and quick method for quantification of the antigen. Okay, the results of rocket immunoelectrophoresis are reproducible. What is meant by reproducible result? That is, if you perform the rocket immunoelectrophoresis experiment multiple times with the same sample, you will going to get similar result. Okay, it is quite accurate method so that its results are reproducible. Then it is the quantitative one-dimensional immunoelectrophoresis technique. Okay, so what is meant by quantitative? The rocket immunoelectrophoresis is mainly used for determining the concentration of antigens. Okay, so it is quantitative method, not a qualitative one. Okay, and it is one-dimensional technique. Why we call it as a one-dimensional? Because at the end of the experiment, we are going to measure the height of the precipitation rockets which is produced in the gel. Okay, so we are going to only focus on the height and not on the area covered by the rocket. So it is one-dimensional approach. Okay, because we are considering only the height and not width of the rocket. Okay, and it is the immunoelectrophoresis. Immuno means the technique involves either antigen, antibody, or any type of immunocomplex. Okay, and it is electrophoresis because we are going to use electric field or electric current for separation or for migration of the antigen in the gel. Okay, so this is the basic of the rocket immunoelectrophoresis. It is the type of a precipitation reaction which is discovered by Laurell. It is simple and a quick method because the method going to give result in 20 instead of 24 hours in 1 hour. Okay, because all other technique require a time of 24 to 48 hours for obtaining the result. But in case of rocket immunoelectrophoresis, you will going to get result in 1 hour. Okay, then it is a reproducible method. It is quantitative and one-dimensional electrophoresis technique. Okay, now we will going to see the requirements. If you want to perform rocket immunoelectrophoresis in your lab, in that case, first you will going to need agarose for forming gels. Okay, so agarose, you can use agarose or agar or polyacrylamide gel. Okay, then in the next requirement is assay buffer. Okay, we know that antigens or antibody are protein and they require specific pH during the reaction mixture, and to maintain the pH we're going to use buffers. Okay, buffer avoid or buffer resists the change in the pH during the reaction so that antigen antibody remain intact. Okay, or they remain functional. So we need assay buffer. Then the third important requirement which one should require during this practical is standard antigens. What is standard antigens? These are the antigen whose concentration is known, okay, before performing the experiment. Okay, so say I'm going to use 4 standard antigen as antigen A, B, C, and D in this experiment. Okay, so the antigen A concentration is say 2 microgram per ml. Antigen B concentration is 4 microgram per ml. Antigen C concentration is 8 microgram per ml. And antigen D concentration is 16 microgram per ml. So I'm going to use 4 different standard antigen whose concentration is known. Then I also need one or many test antigen. What is test antigen? These are the antigen whose concentration is unknown. So aim of the practical or aim of the experiment will be to find out the concentration of this test antigen. At the end, you will going to get concentration of test antigen when you perform rocket immunoelectrophoresis correctly. Okay. The next thing you will need is antibodies or antiserums. These antibodies should be specific for standard and test antigen. Okay. Then next is electrophoresis unit. Because it is a rocket immunoelectrophoresis, so I'm going or we're going to perform electrophoresis of the antigens. So we're going to need electrophoresis unit in the requirements. Okay. Now we're going to see the procedure, step-by-step procedure of rocket immunoelectrophoresis. Once you are ready with all these requirements, you can start the actual procedure. The first step is preparation of 1% gel. So for preparation of 1% agarose gel, you have to weigh 1 gram agarose or agar or polyacrylamide powder, and you have to dissolve that powder in 100 ml assay buffer. So for preparation of the 1% agarose gel, you are going to need these 2 requirements. You have to weigh 1 gram agarose powder and you have to add that 1 gram in 100 ml assay buffer. After addition, the next step will be boiling. Boiling of the above solution to dissolve the agarose in the assay buffer. During the boiling, you will going to see the solution become clear because agarose get dissolved in the buffer. After boiling, the next stage is allow the gel to cool up to 55 to 60 degrees Celsius. Okay, that means you will going to add antibodies directly into the gel, and antibodies are very heat sensitive, and if you add directly this antibody into the boiled agar gel, the antibody get denatured. So after boiling you have to cool the, or you have to wait till the gel get cooled up to 55 to 60 degrees Celsius. And now the next step is, or the fourth step is addition of antibodies. Okay, so now you are going to add the antibodies into the cooled agar gel. After addition, the next step is proper mixing of the antibodies with the gel. Okay, and during the mixing avoid air bubble formation. Okay, and after proper mixing, now you can add, or you can pour this gel onto the glass plate. Pour on glass plate. Okay, so after gel preparation, boiling and addition of the antibody, the next step is, suppose this is the glass plate, okay, which should be clean and dry, and now on this glass plate you will going to pour this gel. Remember that this gel contain antibodies. Okay, so these antibodies are now present in this gel, and you have poured this gel onto the surface of the glass plate. During the pouring, place the glass plate on horizontal surface. Then next step is wait for 30 minutes. Okay, during this period you can keep this plate in a refrigerator to maintain the moisture, and during this 30-minute period the gel gets solidified. Okay, and after solidification of the gel, next step is well formation. You can punch the well, or you can produce the well on this gel at any one side with the help of gel puncture. Okay, so now here you can see, suppose I made 5 different well with the help of gel puncture at one end of this glass plate. Okay, these wells are made with the help of the special type of a gel puncture which avoid the breakage of the well at the time of puncturing. The next step is, after well formation, step number 10 is addition of antigens. Okay, now we have 4 standard antigen with us and 1 test antigen. So total 5 antigen that we have, and now we will going to add these 5 antigens sequentially in this way. Okay, before addition of the antigen, you have to place this plate in the electrophoresis tank. Okay, suppose this is the electrophoresis tank, and now this tank have 2 ends. One end is connected to, say, positive charge anode, and another end is connected to negative charge cathode. Okay, and I have placed this glass plate with the well into this electrophoresis tank, and after that I have to pour electrophoresis buffer in this tank. Okay, so we can pour electrophoresis buffer in this tank. Okay, the buffer should have to cover the gel surface entirely, and then the next step will be the addition of antigens. Okay, you have to add these 5 antigens A, B, C, D and test in the 5 wells with the help of micropipette. Okay, suppose I add antigen A in the first well, B in second, C, D in third and fourth, and test antigen in last well. Okay, and after addition of the antigen in this well, the next step will be application of electric current. Application of electric current. Generally, 100 volts will be applied through this anode and cathode. Okay, because this cathode and anode are directly connected to the power pack. And once you apply the electric current, these antigens are negatively charged. Okay, they are negatively charged. All 5 antigens are negatively charged. So they will going to migrate towards positive anode when electric current is on. Okay, so the antigens start migrating in this direction. Now, during the migration through the gel, the antigen will going to encounter with antibodies which is already present in the gel. We have added antibodies into the gel. So, at the time of migration, this antigen encounter or antigen meet with the antibodies, and due to the antigen-antibody interactions, the precipitation reactions start. We know that in my first video, I have discussed about the principle of the precipitation reaction. When any antigen or antibody meet, they will going to produce 3 different type of zones: pre-zone, zone of equivalence, and post-zone. Precipitation reaction only occur in zone of equivalence. That means for precipitation reaction to occur, the antigen and antibody should be present in equal concentration. Okay, so initially at this point, antigen concentration will be more. Okay, and as antigen move in the gel towards anode, their concentration goes on decreasing. At one point, zone of equivalence reached, and at that point, the antigen combined with antibody to produce insoluble precipitin complex, which will going to give white precipitation line. Okay, so after the electrophoresis, you can see the precipitation line like this. Okay, these lines are obtained or lines are produced in the form of cone or in the form of rockets in the gel. Okay, so this type of precipitation lines are observed. So next step is observation and conclusion. You have to observe the gel carefully, and then you have to make conclusion. I have here drawn rocket with increasing height, because as the height of this rocket is totally depend upon concentration of antigen present in the well. So we know that A antigen have only 2 microgram per ml concentration, so height will be very less. B antigen have 4 microgram per ml concentration, so height will be more. C antigen will going to have 8 microgram per ml antigen, so the height of C precipitation line will be more than A and B, and the highest rocket height will be observed in the well which contain D antigen with 60 microgram per ml antigen concentration. So the height of these rockets which is produced after the electrophoresis is totally depend or totally proportional with concentration of antigen present in the well. The next thing that we have to do is measure the height of every standard and test antigen. Suppose test antigen give rocket of this height, and now we want to determine the concentration of this test antigen which is present in the last well. So now we have to plot a standard graph. Okay, so XY axis of the graph, and on X axis I am going to take concentration of the antigen, and on the Y axis I am going to take height of the rocket. Okay, suppose the concentration is say like 2, 4, 8, 12, 16 and so on. Now we are going to get, when we measure the height of A, B, C and D antigen, and when we plot or when we place the point at 2, 4, 8 and 16 concentration, we are going to get straight line. This straight line or this graph is the standard graph okay, that we have prepared with the help of standard antigen. So the use of the standard antigen is to construct standard graph, and after construction of the standard graph, the last step is measure the height of test antigen rocket, and then just suppose this rocket give a height of say 10 mm. In that case you have to just suppose here on the y-axis it is 10 mm, then you have to just draw a line like this and extrapolate it. Okay, the line touch at the bottom to x-axis, and this point indicate the concentration of test antigen. Suppose it is 8, so now the test antigen will be, or the concentration of test antigen will be 8 microgram per ml. So with the help of standard graph extrapolation, you can easily find out the concentration of test antigen present in the sample. Okay, so this is all about the rocket immunoelectrophoresis. It is the type of a technique which is based upon precipitation reaction, and it is electrophoresis technique, and it is the quantitative method, because with the help of this technique you can easily determine the concentration of antigen, and we called it as a rocket immunoelectrophoresis because the precipitation line form is in the form of cone or rockets. So this line look like rocket, so we call it as a rocket immunoelectrophoresis. So this is all about the precipitation reactions. Okay, we have discussed 4 different techniques or precipitation reaction experiments. Now in my next video I am going to focus on next type of immunoelectrophoresis or immunoreaction, that is agglutination. Thank you.